All right. Well, there, I think we're good there.
So, welcome to the lecture for cell division. We're going to look today at two particular types of cell division. We're going to look at mitosis, and we're going to look at meiosis. Right? These both involve cells, and specifically the genetic contents of cells splitting, but there is a very different purpose behind each of these. So let's go ahead and start off with this beautiful artistic rendering of a man and a woman. Now, I've got here a representative body cell from each individual pictured. The term here is somatic cell. This includes all of your body cells except for egg or sperm cells. So this is going to be all of your cells except for egg and sperm cells. Your brain cells, blood cells, muscle cells, skin cells, they're all somatic cells. And both the man and the woman up here have the number 46 in one of their somatic cells. That 46 represents the number of chromosomes in an individual somatic cell. What's a chromosome? Chromosome is a long strand of DNA with several genes. And I mean, it's a very long strand. It's a wound, tight strand of DNA, and you have many different genes on one single chromosome. What's a gene? A gene is going to be a segment of DNA that codes for a particular trait. So, in other words, you've got these long strands called chromosomes, and each of them contain many different segments that code for particular traits. And they don't even have to be related. On one particular chromosome, there's the genes that determine what sex you are and whether you can perceive red and green colors. So you can have different genes unrelated to each other on the same chromosome. So a chromosome is a really long strand of DNA with several different genes. You have 46 of them. 46 is an even number, and that is for a reason. That's because chromosomes occur in pairs. "23 pairs of chromosomes. 23 times 2 gives you 46. So each of your cells, every single body cell, except for red blood cells, they don't have a nucleus. But other than them, and egg and sperm cells, other than those, all of your body cells have 46 chromosomes, and they exist in pairs. What does it mean for chromosomes to be a pair? Well, this woman and this man each got chromosomes from their biological parents, right? So from her mom and her dad, from his mom and his dad, they received one out of the pair of 2 chromosomes from each parent. 23 chromosomes from her mom, 23 from her dad, same thing with him. Together they made these two with 46 total. So chromosomes occur in pairs, and you get one from each biological parent. So these two each have 46. Well, let's say that they decide to have a child. There is a particular type of cell division that starts with a somatic body cell and trims the number in half from 46 to 23 chromosomes. In other words, only one member of each pair gets passed on. This is going to oversimplify, but let's say there was one gene for eye color. Dad here could have one copy of the gene for eye color that he got from his father that codes for blue eyes and one that codes for brown eyes. He's got both, blue and a brown eye gene. One member of the chromosome pair that has the gene for eye color would have that blue version. The other member of the pair of chromosomes that has the gene for eye color would have the brown version. Chromosomes occur as pairs. When he produces sperm cells, only one member of each pair is passed on. When she produces the egg cell, same thing. Meiosis is cell division that starts off with a cell that has two copies of each chromosome, so the chromosomes occur in pairs, and ends up with new daughter cells that only have one copy. Chromosome number is cut in half. Once his sperm cell and her egg cell fuse, 23, one member of each pair from each parent ultimately results in my clicker stopped working. Oh, there we go. Ultimately results in a single-celled zygote being produced, and this zygote has chromosomes from both the egg cell and the sperm cell. 46 chromosomes, just like both parents. One member of each pair coming from either parent. Over time, that one cell has to develop into trillions more. And each new cell needs to be just like the original in terms of how many chromosomes it has. So, another type of cell division called mitosis is going to keep the chromosome number the same in every single daughter cell as that child will develop into a trillion-celled, well, even as a child there'd be trillions of cells, as a trillion-celled child that came from one individual cell. So, if that didn't make sense, I hope it did. If it didn't, this next part should. This unit will discuss the two major forms of cell division, mitosis and meiosis. So, mitosis starts with a cell that has a certain number of chromosomes. In humans it's 46. And ultimately divides the cell into two daughter cells, each with the same number of chromosomes as what we started with. So mitosis produces two identical daughter cells. It's a lot like asexual reproduction. It keeps chromosome number the same as in the original cell. This is used for growth, repairing damaged cells. For instance, right now if I scrape off some of my skin cells here, I've probably just scraped off several thousand skin cells, my body has to replace those skin cells with new cells that it will build through mitosis. Here's an existing skin cell. Ooh, we need some new ones. Mitosis will produce new identical skin cells, each with the full amount of my chromosomal DNA. Growth, repair, it's also asexual reproduction, right? If you think about how bacteria reproduce, one cell into two cells, identical to the original, mitosis is effectively used in that process. Meiosis, on the other hand, cuts the number of chromosomes in half. Basically, each new daughter cell has one member of each pair. Think about it this way. Think about it like socks. You've got 23 pairs of socks. Actually shoes. Shoes are better. 23 pairs of shoes. 46 total shoes. In mitosis, you have to effectively copy the shoes and end up getting new pairs so that each daughter cell also has 23 pairs of shoes. You got to basically double the amount. In meiosis, you're just splitting each pair of shoes so that one shoe from the pair goes to this cell and one goes to this one. That's ultimately what you're doing with meiosis. So meiosis produces daughter cells with half of the genetic information. And technically, how meiosis works is it's a two-part thing. So you actually end up with 4 daughter cells from the one original. But they all still have just the 23. So that shoe analogy might not have been the best one in that regard. But hopefully it helped you visualize. If you're not familiar yet with the word gamete, gamete is a term that means egg and sperm cells. I just want to quickly mention binary fission. This is cell division in bacterial cells. So binary fission, it's a lot like mitosis in some ways. Bacterial cells have a single strand of genetic material called a plasmid. It's different from your cells. You have chromosomes, right? Several linear strands. A plasmid is a circular strand of DNA that is found in bacterial cells. Binary fission is the asexual reproductive method of bacterial cells. And I want you to notice the most important thing that's happening here. We've got DNA. And the first thing we need to do is duplicate the DNA. Why is that important? I want you to think of your favorite book, right? I really enjoy the Harry Potter series, so let's say that I have the last book, the Deathly Hallows book, and I tell it to my friend, you know, you should read this. And so I want to keep the book for myself, but I also want them to have a copy. So I could cut the book in half. That's not going to work. What I have to do is make a nice illegal photocopy of that and give it to my friend. You wouldn't actually want to do that, but that's how you would have to do it if you wanted both of you to have a copy of the book. You got to photocopy all of the text. So you keep the original and they get the same information that you had. Likewise, we have a cell with all of the DNA. If we just cut this in half and split, one new cell will have the front part of the book, the other new cell will have the back part of the book. That ain't going to work. We need to photocopy the DNA. We have to duplicate it. So that's the next thing, the first thing actually, that's going to happen. Duplicating all of the genetic information. Once that happens, then we can separate those 2 copies of the same genetic information and we split into 2 daughter cells. In bacteria, it's known as binary fission. It's a little bit easier than in your cells because bacteria tend to have 1 circular strand of DNA instead of 46 linear strands like you have. All right? But that's the general idea. Duplicate the genetic information and then split the cell into 2. Sounds good. So let's talk about cell division. You started life as a single-celled zygote. There you are. A zygote has a full copy of all of your DNA. It's got all 46 of your chromosomes. 23 pairs, right? Remember, a chromosome is a linear strand of DNA. Here's a cool picture, which I don't think you can see very well, so let's see if we can magnify this. This is called a karyotype. This is a visual representation of all of your chromosomes. So this individual is a male based on the X and Y chromosomes over here. But notice how each pair consists of the 2 chromosomes. There's 23 pairs, 46 total. Here's a chromosome. Together they are a pair of chromosomes. You had all of those when you were a zygote, a single cell, that formed from the union of the sperm cell and the egg cell that came from your biological parents. You receive 23 chromosomes from your mom, 23 from your dad. Thus, chromosome pairs consist of one maternal and one paternal chromosome. Now, in order for you to grow, that one cell you started at had to duplicate the DNA in it every time it was ready to split again. And we're going to look kind of at how that happens and then how the cell ultimately does split when it's ready to do so. So each new cell also needs to have a full copy of all your DNA. Think of it like this, going back to the book analogy. If one cell is a full copy of a book, all of your cells have to have all of the text for the book. So you consist of trillions of copies of the same book, and that book is your genetic blueprint. But all of your cells have all your genes, which I think is pretty cool. So a little bit more about somatic cells. Outside of red blood cells, all of your somatic cells have all of your DNA. And again, a somatic cell is any cell other than an egg or a sperm cell. For most of the cell's life, your DNA exists as a yarn-like mass in the nucleus of the cell. At that point, it's called chromatin, right? And it's interacting with these things called histone proteins. It's not really nicely ordered in any way. However, before a cell is going to divide, the DNA condenses into the chromosomal shape, which we'll look at. Think of it like this. If you're going to move, you don't want to move every single individual lamp and piece of cutlery or any of that stuff at one individually. You want to move it all at once. And so you put it all into nice, organized boxes and then move the boxes. Makes it a lot easier to do that. When a cell's not dividing, it doesn't really matter if the DNA is in a nice chromosomal shape. Instead, it just exists as a diffuse mass called chromatin. The shape of a chromosome is X-shaped. Okay? So, here's an actual micrograph of a chromosome. What it really would look like, if you were to kind of try and visualize it, is look at the way my marker's going. That is one long strand of DNA. That would be 1/2 of a X shape of a chromosome. Here's the other half. They're so tightly condensed that they make this nice X shape. But again, it's important to note that it's really, really tightly folded in there and compacted quite well. Now I want you to note that there are two halves to this thing. The whole thing is called a chromosome. Each half is called a chromatid. A chromatid is identical to the other chromatid. They are photocopies of each other, mirror images of each other, so to speak. Remember I mentioned if you wanted to give somebody else a copy of your book, you'd have to make a photocopy so you both had full copies? If we were to split the chromosome in half, we've got two copies of the same book connected together at this middle region called the centromere. But sister chromatids are identical. This is not a pair of chromosomes. This involves two copies of the same genetic information linked together. So, this is a pair of chromosomes. Half of one chromosome is called a chromatid. Each of these individually is a chromosome. Together they make a pair. Sister chromatids are identical in every way. They contain 2 copies of the same genetic information from the same parent. Looking at this chromosomal pair, this came from one parent, this came from another. Chromatids on either side of the X, those are identical to each other from the same parent. If you go back, let's go way back. You started off as a single-celled zygote. This zygote has to duplicate. Actually, let's go back even farther. The bacterial cell has to duplicate the genetic information before it can split, so each new cell has all of the genes. Having 2 chromatids is a result of your cell doing what the bacterial cell did and duplicating the genetic information. There was a time in the life of a cell when this is all it had. But then, knowing that it's going to have to split, the cell made an identical copy to that original side, and thus now we have 2 copies of the same thing, so that when we split the cell, each new cell has the full complement of information. Most of a cell's life is spent not dividing, but the cell does divide, and the cell divides during the mitotic or phase, also just called mitosis usually. The yellow and blue part here is interphase. Interphase is the phase that occurs when the cell is not dividing. The 3 stages of interphase, G1, S, and G2. So we've got a cell. Here's our cell, right here. We'll put it actually outside the cycle, right there. During G1, the cell is simply growing. The cell has DNA that occurs with just 1/2 of a hypothetical chromosome. Remember that it's not actually looking like this during this stage, it's still in the chromatin look at this point. But we've got G1, cell's growing, there's 1/2 of a chromosome. That's all we have right now for each chromosome. It looks something like that. In the S phase, that's when the chromosome is going to duplicate and produce the other half of the chromosome. Now we have 2 chromatids. G2 involves additional growth. And then comes mitosis, where the cell is going to ultimately split. Each chromatid, or your chromosomes, going to the new daughter cells. One of them goes off, leaves the cycle. The other one, hypothetically, would go through again and divide again, and that's how you have cell division that occurs over time. The S phase, though, that's when DNA gets synthesized or made, and we duplicate that one chromatid. Let's look at mitosis under a little more detail then. 4 stages occur here: prophase, metaphase, anaphase, telophase. Back when I was in undergrad, one of my fellow students came up with the memory device, pass me a taco, and it's worked for me ever since, and hopefully it'll work for you. But prophase, metaphase, anaphase, telophase, and we're going to look at what happens in each phase. I'm not going to illustrate them on the whiteboard, though. We're going to look here. So this is prophase. This is right when mitosis is about to begin. Things to note. The nucleus is still intact. The nuclear envelope, that's the outer membrane of the nucleus, is still intact. By this point, DNA has condensed into the chromosomes. It's not in the chromatin stage still. This particular individual has 6 chromosomes. I didn't feel like illustrating 46, which is 3 pairs. What makes them a pair? Well, these guys are a pair, these guys are a pair, and these guys are a pair. What makes them a pair is that they're the same length, same shape. The color does not make them a pair. The color represents which parent this hypothetical individual got them from. The red chromosomes would have come from their biological father, the yellow ones from their biological mother. And notice how for each pair of chromosomes, they have one from each parent. So we have chromosomes condensed inside the nucleus. Out here in the cytoplasm, we also have this thing called the mitotic spindle, which is built from microtubule protein fibers. It's starting to form. And there are these 2 things called centrosomes. In animal cells, they have something called centrioles. I'm not too worried about that for you guys. But this mitotic spindle is starting to form, and it's going to be pretty darn important in a little bit. So keep this in the back of your mind. But in prophase, really what we see is that chromosomes are starting to condense in the nucleus of the cell. It's also important to note that chromosomes are not associating as a pair. You may have seen this word before, but it should be new for this PowerPoint. Chromosomes that are homologous are members of a pair. So homologous means coding for the same thing. So if you have a pair of chromosomes, one you got from your mom, one you got from your dad, and what makes them a pair is that on the same spot in each chromosome, you've got a gene that codes for the same thing. Doesn't have to be identical, but it codes for the same thing. For instance, it's not this simple, but let's pretend it was. There's a gene for the color of your eyes. On both members of the homologous pair of chromosomes, you've got that gene for eye color in the exact same spot on the one you got from your dad and the one you got from your mom. Meanwhile, down here, on a different part of that same chromosome, there might be a gene that codes for how tall you are, or something along those lines. Point is, you've got genes that code for the same stuff in the same places on chromosomes that are members of a homologous pair. So that's what it means to be a pair of chromosomes. Notice how homologous chromosomes are not associating with each other during mitosis here. They're just not. This one and this one are homologous pairs and they're nowhere near each other. During mitosis, homologous pairs don't associate with each other at all. They just don't have to. And so all 46 of your chromosomes are just scattered throughout the nucleus. The second phase is metaphase. So this phase is metaphase. And this involves the chromosomes aligning at the equator. I like to think of this as the planet Earth. And so the chromosomes are aligned at the equator. Notice that the centrosomes are now at the poles. Think of it like the Earth, North and South Pole. The microtubule protein fibers have attached here in the cell to the chromosomes, as well as they're still attached to the centrosomes. But let's take a look at the chromosomes here. Notice how they've aligned themselves at the equator. I know there's a better term for this, but I still don't know it. They're in a single file line. They're in a single line. They're not paired up. They're not arranged like this with two of them. Nope, they're in a single file line. In your cell, this would be 46 chromosomes long. Also notice that homologous chromosomes are not paired up or with the other member of their pair whatsoever in here. It's just random chance how they align themselves. If you think of it like shoes, it would be as if you had one boot here and another boot happened to be here. This one would be a pair of slippers. Here's the other shoe that's a slipper, and these two happen to be tennis shoes. It's just randomly arranged. They're not put anywhere near the other half of that pair. So chromosomes align in a single file line at the equator. The centrosomes are at the poles of the cell. These lines are microtubule protein fibers. They are excreted by the centrosome and they attach to the chromosomes. If you were to magnify this, it would look something like this. The microtubules coming from the North Pole centrosome would latch on to the top chromatid, and the microtubules coming from the South Pole centrosome would latch on to the bottom chromatid. The technical name for where they latch on is called the kinetochore, and basically, if you can visualize it, the microtubules are going to shorten and pull the chromatids apart. That's exactly what happens in the next phase if you just watch the visual here. We are pulling chromatids apart. This is anaphase. In anaphase, sister chromatids are pulled apart from each other as the centrosomes, for lack of a better way of phrasing it, reel them in toward themselves. The microtubules shorten, pulling the chromatids toward the centrosomes. The cell itself is also elongating, so it's getting ready to ultimately split. So we're going to separate effectively along that line. So this is anaphase. Telophase is the last phase of mitosis, and it involves the splitting of the cytoplasm, which is called cytokinesis. And by the time we get here, we've got two daughter cells about to form. Now, that each of these half chromosomes is fully capable of duplicating itself so that you can think of it basically as having the same contents as a full chromosome. It's like it has one copy of the book instead of two, that's all. The point being, each new daughter cell still has two copies of each chromosome. I messed that up. There we go. Each daughter cell has two copies of each chromosome, just like the cell that we started with. The only difference is that the daughter cells haven't yet gotten to the point where they're going to duplicate the DNA and split themselves. But they've got two copies of each chromosome, the full content of genetic information, like we started with. We've made, effectively, clones of the one original. And that's the whole point. This last phase is called telophase. Here's a written summary of what happens in each phase. I recommend you read through that. Typically, the phrase mitosis refers just to cell division involving the genetic contents of the cell. It doesn't actually refer to the full splitting of the cell, including the cytoplasm and all of that. Cytokinesis is the division of the cytoplasm. That's the semi-fluid interior of the cell. In animal cells, cytokinesis occurs through the formation of a cleavage furrow, which is this thing here, which effectively squeezes the cell into two daughter cells. I want you to imagine as if there was a piece of string around this and you just pulled it and squeezed the cytoplasm and separated the two daughter cells. That is cytokinesis in essence. Plant cells, that doesn't work very well for cytokinesis. They have a cell wall. So this cleavage furrow strategy is not going to cut it. So they have to do something different. In plant cells, they just build a new cell wall. So here we have a couple of plant cells. These are the two new genetic components. I want to say that the nucleus has started to reform. So these are the two new nuclei of the cell. This is the starting construction, is that the right word? The plant's construction of a new cell wall to separate what was one cell into two. Basically, we started with one plant cell. There's the nucleus. We duplicated the two nuclei, that. And then we separate it through the formation of a cell wall. That's how plant cells do it. So a quick summary of mitosis. We started off with cells that had 2 copies of each chromosome. In humans, that would be 46 or 23 pairs. And we ended up with 2 daughter cells that had exactly the same number of chromosomes. The word of the day is diploid. Di means 2. A cell is diploid if it has 2 copies of each chromosome. All of your somatic cells, if you remember that word means body cells. All of your somatic cells are diploid. They have 2 copies of each chromosome. Oftentimes this is represented as 2n. N simply equals the number of chromosomes. Different species have different numbers of chromosomes. A lot of species have fewer than we do, a number of species have more. But if they have 2 copies of each, they're said to be diploid and thus have 2n. In humans, n equals 23. So 2n, 2 times each chromosome is 46. Here's a nice summary picture. I'm not going to dwell on it too much, but you can pause the video if you want, take a look. It talks about the original cell and the daughter cells of mitosis.